एकदा दिसली एकदा.
Hello everyone, I am Arushi and I am the house captain of Harvey house. Here are my house members. Here we have made a model about stages of fertilization for our inter-house competition on embryology model making, that is initial. Hello everyone, I am Arushi and here we have a model of stages of fertilization. So let's begin. Fertilization is the process by which male and female gametes fuse to give rise to a diploid zygote, and human fertilization occurs in the ampullary region of the fallopian tube. Here we have the structure of the sperm or a spermatozoa. The sperm can be briefly divided into 4 parts. First we have head, then neck, then we have middle piece, and then principal piece. The head contains the acrosome, which is formed by the Golgi apparatus. It contains various enzymes such as acrosin, hyaluronidase and pepsin for penetrating the secondary oocyte. Then we have nucleus. The chromatin material inside the nucleus is highly condensed. And then we have neck. Neck is very narrow and has a basal body and a centriole. The centriole gives rise to an axial filament which extends into the tail. And we have mitochondria which lie in the middle piece of the spermatozoa. The mitochondria are also known as strips of a Leverkusen, which provide energy for the motility of the sperm. And then we have a long tail, which also helps in mobility of the sperm. Here we have structure of an ovum or a secondary oocyte. The outermost layer of the secondary oocyte is formed by corona radiata cells which stick out from zona pellucida layer of the secondary oocyte. Then we have perivitline space which is clearly visible, and it has a first polar body in it. Then we have nucleus. The nucleus doesn't have a nuclear membrane which is shed during the first meiotic division. There are a number of cortical granules which lie in the ooplasm, and also we have mitochondria lying in the ooplasm.
I am Harsha, I am from Harvey group. Now I am going to explain about the stages of fertilization. Actually, during the coitus, 200 to 300 million sperms are released in the vagina, but only 200 to 300 sperms will reach the ampullary region of the uterine tube, so that we can estimate the competition existing in the world from our childhood and birth. So actually, when we discuss about the journey of the sperm from the vagina into ampullary region of the uterine tube, initially it starts by secretion of prostaglandins from the semen, which will help in guiding the route of the sperm up to the cervix. And in the cervix there will be the self-motility of the sperm from the tail, which is promoting the movement. And again the movements like fluid currents which are generated by the uterine cilia will help in sucking of the sperm, and which will create a negative pressure. And then it will move upward near the ampullary region. There will be secretion of a chemoattractants from the cumulus cells surrounding the oocyte, so that the sperm will become motility, and as well as it come and fertilize that. And then if we move to the major topic, which is very important reactions, the prerequisites for fertilization. The first one is the capacitation. In this, the conditioning and maturation of the sperm occurs, and it lasts about the 7 hours after the coitus. And as well as if we go through the topic, we have 2 major steps occurring here. The removal of the glycoprotein coat from the head of the sperm, and as well as removal of the semen plasma proteins from the sperm. And this makes the sperm ready. And then when we move to the second important reaction called acrosomal reaction, when the capacitated sperm comes in contact with the zona pellucida, there will be release of enzymes called hyaluronidase and as well as acrosin enzymes. And also this will lead to some immunological reaction by the sperm anti-fertilizin and as well as ovum fertilizin. So the clinical correlation for this prerequisites is nothing but capacitation is very important step, and which lasts about 7 hours, and it is very mandatory for a sperm to become capacitated and undergo priming. And then if we move on to the next topic, the important topic, the stages of fertilization. The first, we have 3 barriers around the secondary oocyte. The first one is the corona radiata, and inside that there is zona pellucida, and inside that there is a white line membrane. The cleavage of these 3 barriers will lead to the fertilization of the sperm with the secondary oocyte. So when we move on to the first step, which is the important step, which leads to the penetration of corona radiata by the release of hyaluronidase enzyme from the acrosomal cap of the sperm, so that there will be penetration of the head of the sperm into the secondary oocyte. And then if we move on to the second reaction, the sperm enters into the second layer, which is a zona pellucida layer that contains the glycoprotein receptors called ZP2 and ZP3. The sperm acrosomal cap have these glycoprotein coats which will interact with those receptors, and as well as pepsin comes and cleaves them, so that the sperm enters, leaving the plasma membrane behind, and as well as the neck and tail behind. And then when we move on to the third topic, and when we move to the third one, which is the fusion of the Whitelane membrane. When the sperm enters the Whitelane membrane, it produces a calcium wave. The calcium wave is very important because it undergoes a Whitelane depolarization. Actually, the cortical granules which are present in the center of the cytoplasm of the ovum will split into the periphery, so that there will be no polyspermy entering and only one sperm will enter, so that the hardening of the Whitelane membrane and as well as zona pellucida takes place, which will prevent the polyspermy. And then when we move on to the fourth step, which is very important step, there will be formation of male pronuclei. The male pronuclei when enters, the male head of the sperm enters and it becomes swollen, and it will readily fertilize with the secondary oocyte. And then the fifth step is nothing but the formation of female pronuclei, so that when male pronuclei comes and fuses with that, there will be completion of second meiotic division of the secondary oocyte, so that there will be production of two cells. The first cell is which contains highest cytoplasmic region, and other one which contains lowest cytoplasmic region. The highest cytoplasmic cell converts into a mature oocyte, and again the one which contains less cytoplasm will convert into a polar body, and it will be left in the peri-Whitelane space, so that the large one which developed as a mature oocyte will undergo further changes and will promote the metabolic changes in the cleavage. And then when we move on to the results of the fertilization, the first one which occurs is the determination of the sex at the chromosome level, and the second one is restoration of the diploid sex of the chromosome, which is very important to maintain the generations, and as well as variation of the species is very important because sexual reproduction occurs only by this process. And then in the process of fertilization, if two X chromosomes are present in a person, this is termed as a female, and one X, one Y chromosome will be termed as a male. Oftenly in more regional institutions or hospitals, all will be blaming the female for having the child birth for a male or female, but it is mandatory to know the science behind it. Only the male is responsible for the release of X or Y chromosome, and the female is not blamed upon this issue. And then when we move on to the clinical correlation of this topic, it is in vitro fertilization. In vitro fertilization, the main step is hormonal stimulation by the prostaglandins and progesterone, which will stimulate the follicles to produce more oocytes. And in the second step, we will collect the oocytes from the Graafian follicles, and again we will store them in a lab, and again we will collect the semen and make them capacitation, as it is the major step for getting fertilized. In the fourth step, we will make these to fertilize in a culture medium by artificial enzymes, by acrosin peptidase enzymes. And then when we come to the fifth step, it is grown up to the 8 cell stage, and in the 8 cell stage it is re-employed into the uterine tube so that it can develop. Actually, this process is employed because the male couldn't ejaculate the number of sperms, and as well as due to some rupture in the fallopian tube or in issues during the implantation. So we should have some moral support and social stigma sticking in it, because so many people will not have the babies, but that may be due to the natural, physical or chemical conditions, but we should not blame them, and we should give them the moral support in having the babies in that manner. Who doesn't have, they will also have some feelings towards them. So don't ever blame the persons who have undergone in vitro fertilization, as science is a part of life.
3.
A very big thanks to Dr. Chitra ma'am for giving us this opportunity to present our model on stages of fertilization.
Thank you for giving us this opportunity, Chitra ma'am, to present the embryological models of fertilization. Thank you.